press the bell icon on the YouTube app and never miss another update.
Hello students, in last video we discussed about the development of endosperm. So in angiosperm it is an adaptation where the development of endosperm occurs first when compared to the development of embryo, that is by process called as embryogenesis. So before embryogenesis the endosperm formation occurs. It is an adaptation in the angiosperm to nourish the developing embryo. Okay. So in this class we study about embryogenesis. What is this embryogenesis? It is nothing but the process of development of embryo from zygote. Process of development of embryo from zygote. See here, this is the zygote. Universally zygote is diploid and the zygote is single celled. This zygote is single celled. It is made up of single cell. Best example for largest single cell zygote is the egg. So here the embryo is this here. These are the embryos. These embryos are multicellular in structure, but zygote is single celled, and both the zygote and embryo are diploid in nature in angiosperms. So this single celled zygote consists dense cytoplasma and the nucleus. So this is the cytoplasma and this is the nucleus. Okay. Then this zygote undergoes mitotic division. The first mitotic division leads to formation of primitive embryo or first embryo, which is made up of two cell structure called as proembryo. When the zygote divides mitotically, it gives rise to two cells, and the two celled embryo is called as proembryo. Next, this proembryo undergoes repeated mitotic division. Here the mitosis, or what we call as cleavage. Here what happens, that immediately after karyokinesis the cytokinesis occurs. Okay, karyokinesis is followed by cytokinesis and it leads to formation of globular embryo. See the proembryo develops what? A globular embryo, a globe shaped embryo which consists the ostomium. The ostomium is fixed. It fixed the embryo inside what? Inside the micropylar region. So the ostomium fixed the embryo inside the embryo sac in micropylar region. See, this is the micropylar region. These are chalazal end. So the ostomium, it firmly fixed the embryo in micropylar region, and where the other parts of embryo grows towards chalazal end. Then this cells, this elongated cells is called as suspensor. Okay. Then this is the globular embryo, that is globe shaped embryo which consists many number of cells and nucleus. Next, the globular embryo, it gives rise to an heart shaped embryo. odd shaped embryo. See here, globular embryo develops into an odd shaped embryo. The embryo looks like odd like structure, and here it consists the ostomium and the suspensor. Here also the ostomium is firmly fixed inside the embryo sac towards micropylar region, and the embryo is growing inside the embryo sac towards chalazal end. Then this odd shaped embryo further give rise to a matured embryo. Odd shaped embryo further give rise to matured embryo, which also consists the ostomium and suspensor and the fully formed embryo. Okay, so see here, the zygote is single cell formed by the process of fertilization towards micropylar end. This zygote undergoes first cleavage or mitotic division to form two-celled embryo called as proembryo. Proembryo undergoes further mitotic division to form a shape called as globular shaped embryo, which consists the ostomium and suspensor. Ostomium is firmly fixed inside the embryo sac towards micropylar region, and other parts of embryo is developed towards chalazal end in the embryo sac. Next, the globular embryo, after it formation, it also undergoes continuous division to form the odd shaped embryo. Then odd shaped embryo further develops into matured embryo. So still for development of zygote into matured embryo is similar in both dicots and monocot. ಅಂದರೆ ಝೈಗೋಟ್ ಇಂದ ಮೆಚ್ಯೂರ್ ಎಂಬ್ರಿಯೋ ಫಾರ್ಮೇಷನ್ ಆಗೋಗ್ಬೇಕಾದ್ರೆ ಅದೇನು ಪ್ರೋಸೆಸ್ಸು ಮತ್ತು ಶೇಪ್ ಇದೆ ಅದು ಒಂದೇ ರೀತಿ ಇರುತ್ತೆ. ಯಾವುದರಲ್ಲಿ ಬೋತ್ ಡೈಕಾಟ್ಸ್ ಮತ್ತು ಮಾನೋಕಾಟ್ಸ್. ಫರ್ದರ್ ಡೆವಲಪ್ಮೆಂಟ್ಸ್ ಫ್ರಮ್ ಮೆಚ್ಯೂರ್ ಎಂಬ್ರಿಯೋ ಇಂದ ಫರ್ದರ್ ಇನ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗುತ್ತಲ್ಲ, ಅದು ಆವಾಗ ಏನಾಗುತ್ತೆ, ನಮಗೆ ಡೈಕಾಟ್ ಎಂಬ್ರಿಯೋ ಶೇಪೇ ಬೇರೆ ಇರುತ್ತೆ, ಮಾನೋಕಾಟ್ ಎಂಬ್ರಿಯೋ ಶೇಪೇ ಬೇರೆ ಇರುತ್ತೆ. So now we should discuss both the dicot embryo, shape of dicot embryo and shape of monocot embryo. Okay, here see this shape is for dicot embryo. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? ಸೊ ಇಲ್ಲಿ ತನಕ ಏನು ಎಂಬ್ರಿಯೋಜೆನೆಸಿಸ್ ಇರುತ್ತೆ, ಇಟ್ ಇಸ್ ಸಿಮಿಲರ್ ಇನ್ ಬೋತ್ monocot and dicot. After this the further development is give rise to different shape, that is one dicot or another type monocot. See, the dicot embryo consists two cotyledons, so the name is called as dicot, two cotyledons. Okay, then the upper part of the embryonic axis is called as epicotyl, and the epicotyl give rise to plumule. Plumule further develops into shoot system. Then the lower part of the embryonic axis is called as hypocotyl. The hypocotyl give rise to radical. Hypocotyl give rise to radical. Radical further develops into root system. Okay, so the dicot embryo consists two cotyledons and the embryonic axis. The upper part of the embryonic axis is called as epicotyl, which give rise to plumule. Plumule further develops into shoot system. Then lower part of the embryonic axis is called as hypocotyl. Hypocotyl develops into radical. Radical further develops into root system. So this is the shape of or structure of dicot embryo. Next, the monocot embryo. The mature embryo develops into monocot. Then monocot embryo reveals the following structure. It contains the endosperm, endosperm is preserved in monocots. So, this majority of monocots are endospermic seed. This is an example for monocot, that is the grass. Okay. Then the single cotyledon is called as scutellum. Scutellum is the single cotyledon of monocots, is called as scutellum. Then the upper part of the embryonic axis is called the plumule. But the plumule here is covered by an extra protective sheath because it contains only a single cotyledon, the shield shaped cotyledon called as scutellum. Then the plumule is exposed, so the plumule is protected by a sheath called as coleoptile. The sheath is called as coleoptile, which protects the plumule. Then lower part of the embryonic axis is called as radicle. Radicle is protected by a sheath, a protective sheath called as coleorhizae. Radicle is covered by the protective sheath called as coleorhiza. Then plumule is covered by protective sheath called as coleoptile. Next, see here, a structure, a rudimentary structure is present, is called as epiblast. It is called as epiblast. It is nothing but the secondary cotyledon. Epiblast is called as secondary cotyledon. Secondary or rudimentary cotyledon, very important for competitive exam. Epiblast is called as rudimentary, ಅಂದರೆ ಅದು ಗ್ರೋತ್ ಆಗ್ತಾ ಇರೋದಿಲ್ಲ. ಇವಾಗ ನಮ್ಮಲ್ಲಿ ಹಿಂಗೆ ಇಯರ್ ಪಿನ್ನ rudimentary ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಕರಿತೀವಿ, ಅಪೆಂಡಿಕ್ಸ್ rudimentary ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಕರಿತೀವಿ. ಅಂದರೆ ಅದು ಡಿಜನರೇಟ್ ಆಗ್ತಾ ಇದೆ, ಗ್ರೋತ್ ಆಗ್ತಾ ಇಲ್ಲ. ಸೆಕೆಂಡ್ ಅಂದರೆ ಎರಡನೇ ಕಾಟಿಲಿಡನ್, ಸಿಂಗಲ್ ಕಾಟಿಲಿಡನ್ ಮಾತ್ರ ಇದೆ, ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ as scutellum. ಓಕೆ, ಸೊ ದಿಸ್ ಈಸ್ ದ ಎಕ್ಸಾಂಪಲ್ ಫಾರ್ monocot embryo, that is grass. Example, grass. 2 ಮಾರ್ಕ್ಸ್ಗೆ ಕೇಳ್ತಾರೆ ಗ್ರಾಸ್ ಒಂದನೇ, 5 ಮಾರ್ಕ್ಸ್ಗೆ ಎಂಬ್ರಿಯೋಜೆನೆಸಿಸ್ನ ಕೇಳ್ಬೋದು. ಓಕೆ, ಸೊ development of zygote to mature embryo is similar in both monocot and as well as in dicot. Further differentiation of mature embryo occurs differently in dicot and in monocot. In dicot, it gives rise to 2 cotyledons and a single embryonic axis. The single embryonic axis consists plumule and radicle. In monocot, it is a single cotyledon called as scutellum, and where it contains plumule and radicle, but plumule is covered by a protective sheath called as coleoptile, and radicle is covered by a protective sheath called as coleorhiza. Okay, so in the ಬರ್ಕೊಡಿ diagram important. So we discussed about the process of or events of fertilization. In last video I told you the events of angiosperm fertilization is divided into pre-fertilization, fertilization and post-fertilization. In post-fertilization we studied development of endosperm and finally development of embryo. So after fully development of embryo, now that structure where the embryo is developed, the embryo is developed inside the embryo sac of ovule and ovule is present inside the ovary. Now after development of embryo and after the process of fertilization, the ovary also grows, ovule also grows. Then the ovary now is called as fruit. The ovary now we called as fruit. What is fruit? Fruit is nothing but fertilized, matured, ripened ovary. Fruit is a fertilized, matured, ripened ovary. Fertilization ಆಗಿರುತ್ತೆ, ಮೆಚುರಿಟಿ ಆಗಿರುತ್ತೆ, ಹಣ್ಣಾಗಿರುವಂಥ ಓವರಿನ ನಾವೇನಂತ ಕರಿತೀವಿ? ಫ್ರೂಟ್ ಅಂತ ಕರಿತೀವಿ. Then the fruit is fruit wall. Fruit wall is called pericarp. Fruit wall is called as pericarp. This pericarp is divided into 3 layers, that is outer skin called as epicarp, outer skin called as epicarp, middle part we called as mesocarp, then innermost layer we called as endocarp. Totally we called as pericarp. It's nothing but the fruit wall. Okay, so when you take the fruit like mango, here we can easily differentiate these 3 layers. Outermost skin of mango is called as epicarp, then middle juicy layer is called as mesocarp, then inner odd layer is called as endocarp. Totally we called as pericarp. This is nothing but fruit wall, right? Next, types of fruit. So fruit is having following types. True fruit is a fruit developed from ovary. If ovary develops into fruit, then it is called as true fruit. Then second type, that is false or pseudocarpic or false fruit. Pseudocarpic or false fruit. Here the fruit developed other than ovary. If the fruit develops other than ovary, from other parts of the ovary, like especially like thalamus, like thalamus, then that fruit is called as pseudocarpic fruit or false fruit. Thalamus is also called as receptacle or thorus. So it is the tip of the pedicel. The tip of the pedicel. Pedicel means stalk of the flower. The tip of the pedicel is somewhat flattened, cup shaped or disc shaped structure is called as thalamus. If thalamus develops into fruit, then it is called as pseudocarpic or false fruit. Example, apple, then cashew nut alla, cashew fruit, okay, cashew fruit ಏನಿದೆ ಅದು. ಸೊ ಇವೆಲ್ಲ ಏನಪ್ಪ ಅಂತಂದರೆ ಫಾಲ್ಸ್ ಫ್ರೂಟ್ ಅಂತ ಸೇರಿಸ್ತೀವಿ. ಪೀರ್ಸ್, ಸೊ ಅವನ್ನೆಲ್ಲ ಫಾಲ್ಸ್ ಫ್ರೂಟ್ ಅಂತ ಕರಿತೀವಿ. ಟ್ರೂ ಫ್ರೂಟ್ ದಟ್ ಈಸ್ ಮ್ಯಾಂಗೋ, ಕೋಕೋನಟ್, ಲೆಮನ್, ಆರೆಂಜ್, ಕುಕುಂಬರ್, ಟೊಮ್ಯಾಟೊ, ಸೊ ಅವನ್ನೆಲ್ಲ ಟ್ರೂ ಫ್ರೂಟಿಗೆ ಬರುತ್ತೆ. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಟೈಪ್ ಆಫ್ ಫ್ರೂಟ್ seedless fruit or parthenocarpic fruit. Parthenocarpic fruit. It is the fruit developed from ovary but without fertilization. If the fruit developed from ovary without fertilization, here ovary or ovules does not undergo fertilization, so where seed formation is absent, only fruit formation is occur. Example, natural parthenocarpic fruit is banana and some grapes. But nowadays, by using the auxins and other growth promoters, so different fruits are found into parthenogenesis where the seeds are not required, like lemon, cucumber. So those fruits are made into parthenogenesis where seeds are not eaten by the peoples. Okay, so these are the main types of fruit: true fruit, pseudocarpic fruit, and parthenocarpic fruit. Then other type of fruits are also there, and it is studied in first year. That is simple fruit, dry fruit. Okay, means fleshy fruit, dry fruit. In simple fruit, that is fleshy fruit. Then dry fruit, dry dehiscent fruit, dry indehiscent fruit. Then aggregate fruit and multiple fruit. It is well studied in the first year PUC. Okay, take down this. So we studied about fruit. Next is seed. The definition for seed: seed is fertilized, matured, fertilized, matured, ovule is called as seed. So after fertilization, ovary develops into fruit, ovule develops into seed. Next, the part of seed. See here, the seed, okay, a seed, a single seed is divided into two parts. First, we called as seed coat. Seed coat. It is the outermost part of the seed. The seed coat is divided into two layers. The outermost layer is called as testa. Then inner layer is called as tegmen. Testa and tegmen. Testa is outermost layer, and tegmen is innermost layer of the seed. Next, embryo. Embryo is divided into two parts, that is cotyledons and embryonic axis, right? Cotyledon based on number of cotyledon is single or it is two, then it is called as dicots. Is single, then it is called as monocots. Cotyledons in seed, if cotyledons is two, then it is dicots. Is one, then it is called as monocots. Then the embryonic axis, the upper part of the embryonic axis is called as plumule. Plumule is the upper part of embryonic axis. Then lower part of embryonic axis is called as radicle. Radicle is the lower part. You know that the plumule give rise to shoot system, radicle give rise to root system. So these are the parts of a seed. Parts of a typical seed. So seed coat and embryo. One seed, dicot seed, ಈ ಶೇಂಗಾ ಬೀಜ ಇರ್ತಲ್ಲ ಗ್ರೌಂಡ್ನಟ್ ಸೀಡ್ ನೋಡಿದ್ದೀರಾ ನೀವೆಲ್ಲ ಅಲ್ವಾ? ಸೊ ಅದು ನೋಡೋ ಸಾಕು ಈಸಿಲಿ ಗೊತ್ತಾಗುತ್ತೆ. ಔಟರ್ ಮೋಸ್ಟ್ ಸೀಡ್ ಕೋಟ್ ಇರುತ್ತೆ. ಸೊ ಅದು ಟೆಸ್ಟಾ ಟೆಗ್ಮಾ ಅಂತ ಹೇಳ್ತೀವಿ. ದೆನ್ ಎಂಬ್ರಿಯೋ ಇರುತ್ತೆ, ಎಂಬ್ರಿಯೋದಲ್ಲಿ ಕಾಟ್ಲಿಡನ್ ಇರುತ್ತೆ. ಕಾಟ್ಲಿಡನ್ನ ಸ್ಪ್ಲಿಟ್ ಮಾಡಿದ್ರೆ ಬುಕ್ ಥರ ಓಪನ್ ಆಗ್ತಾ ಇರೋ ಒಂದು ಮೂಲೆಯಲ್ಲಿ ಪ್ಲ್ಯೂಮ್ಯೂಲ್ ಮತ್ತು ರ್ಯಾಡಿಕಲ್ಸ್ ಕಾಣಿಸ್ಕೊಂಬತ್ತೆ. ಸೊ ಲೀಫಿ ಲೇಯರ್ ಇರುತ್ತಲ್ಲ ಅದು ಪ್ಲ್ಯೂಮ್ಯೂಲ್. ದೆನ್ ಕೆಳಗಡೆ ಬಲ್ಬಸ್ ಶಾರ್ಪ್ ಕೆಳಗಡೆ ಇರುತ್ತಲ್ಲ ಅದು ಪಾಯಿಂಟೆಡ್ ರ್ಯಾಡಿಕಲ್ ವಿಚ್ ಡೆವಲಪ್ಸ್ ಇಂಟು ರೂಟ್ ಸಿಸ್ಟಮ್. ಸೊ ದಿಸ್ ಇಸ್ ದ ಪಾರ್ಟ್ ಆಫ್ ಸೀಡ್. ಬರ್ಕೊಳ್ಳಿ ಇದು. Next, types of seed based on endosperm. Types of seed based on presence or absence of endosperm. So there are two types of seed, that is non-endospermic or X albuminous seed. Based on presence and absence of endosperm, there are of two types of seeds. One we called as non-endospermic or X albuminous seed. These are the seed where developing embryo, during the process of embryogenesis we studied the development of embryo. Okay, during the development of embryo, these are the seed where developing embryo if completely consume the endosperm and the seed does not consist the endosperm, then that seed is called as non-endospermic seed or X albuminous seed. ಅಂದರೆ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತಿರುವಂಥ ಎಂಬ್ರಿಯೋ ಆ ರಿಸರ್ವ್ ಫುಡ್ ಮೆಟೀರಿಯಲ್ಸ್ ಏನು ಎಂಡೋಸ್ಪರ್ಮ್ ಇರುತ್ತೆ ಅದನ್ನು ಕಂಪ್ಲೀಟ್ಲಿ ಯೂಸ್ ಮಾಡ್ಕೊಂಡ್ಬಿಡುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಅದು ಫ್ಯೂಚರ್ಗೆ ಅಂತ ಇಟ್ಕೊಳ್ಳೋದಿಲ್ಲ. ಸೊ ಅವಾಗ ಆ ಸೀಡಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದೇ ಇಲ್ಲ, ಕಂಪ್ಲೀಟಾಗಿ ಅದು ಯುಟಿಲೈಸ್ ಮಾಡ್ಕೊಂಬಿಟ್ಟಿರುತ್ತೆ. ಅಂಥ ಒಂದು ಸೀಡನ್ನು ಏನಂತ ಕರಿತೀವಿ, ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ ಆರ್ ಎಕ್ಸ್ ಆಲ್ಬ್ಯುಮಿನಸ್ ಸೀಡ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಎಕ್ಸಾಂಪಲ್ ಆರ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ನೀಟ್. See, definition are important for theory exam. Example, majority of dicots are non-endospermic seed like pulses. Example, gram, groundnut, pea, beans. ಓಕೆ, ಸೊ ಏನು ನಾವು ಪಲ್ಸಸ್ ಅಂತ ಕರಿತೀವಿ, ಆ ಪಲ್ಸಸ್ಸಲ್ಲಿ ಮತ್ತೆ ಮೆಜಾರಿಟಿ ಆಫ್ ಡೈಕಾಟ್ಸಲ್ಲಿ ಏನಿರೋದಿಲ್ಲ, ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದಿಲ್ಲ. ಸೊ ಇಂಥದ್ದ ಒಂದು ಸೀಡ್ಸ್ನ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ ಆರ್ ಎಕ್ಸ್ ಆಲ್ಬ್ಯುಮಿನಸ್ ಸೀಡ್ ಅಂತ ಕರಿತೀವಿ. Then monocots ಅಲ್ಲಿ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್. ಎಕ್ಸಾಂಪಲ್, ಆರ್ಕಿಡ್ಸ್ ಲೈಕ್ ವೆನಿಲಾ ಎಲ್ಲ ಇದೆಯಲ್ಲ, ಸೊ ಆರ್ಕಿಡ್ಸ್ ಅಲ್ಲಿ ಅದು ಪ್ರಿಮಿಟಿವ್ ಫ್ಯಾಮಿಲಿ ಆಫ್ ಮೊನೊಕಾಟ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಅದರಲ್ಲೂ ಕೂಡ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದಿಲ್ಲ, ಬಟ್ ಮೆಜಾರಿಟಿ ಆಫ್ ಮೊನೊಕಾಟ್ಸ್ ಆರ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್. ಓಕೆ, ಸೊ ಈ ಮೊನೊಕಾಟ್ಸ್ ಲೈಕ್ ಆರ್ಕಿಡ್ಸ್ ಅಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದಿಲ್ಲ, ಇದು ಕನ್ಫ್ಯೂಷನ್ ಆಗುತ್ತೆ. ಸೊ ಹಾಗಾಗಿ ನೀಟಾಗಿ ನೋಡ್ಕೋಬೇಕು. ಎಕ್ಸಾಂಪಲ್ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ದಿಸ್ ಈಸ್ ದ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್. ದೆನ್ ನೆಕ್ಸ್ಟ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ ಬರ್ಕೊಳ್ಳಿ. ಇದನ್ನು So endospermic seed or albuminous seed, these are the seed where developing embryo does not completely consume the endosperm and the seed concedes and the seed preserves or the seed preserves or concedes the endosperm, then it is called as endospermic or albuminous seed. ಡೆಫಿನೇಷನ್ ಎರಡು ಕೂಡ ಒಂದೇ ಥರ ಇರುತ್ತೆ. ಅಲ್ಲಿ ಸೀಡಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದಿಲ್ಲ, ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕಲ್ಲಿ. ಇಲ್ಲಿ ಸೀಡಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಉಳಿಸ್ಕೊಳುತ್ತೆ. ಡೆವಲಪಿಂಗ್ ಎಂಬ್ರಿಯೋ ಡಸ್ ನಾಟ್ ಕಂಪ್ಲೀಟ್ಲಿ ಕನ್ಸ್ಯೂಮ್ ಮಾಡ್ಕೊಳಲ್ಲ ಅಂತ ಮಾಡ್ಕೊಳುತ್ತೆ, ಆದರೆ ಒಂದು ಹಾಫ್ನ ಇಲ್ಲಾಂದರೆ 75% ಪ್ರಿಸರ್ವ್ ಮಾಡ್ಕೊಳುತ್ತೆ ಫಾರ್ ಫ್ಯೂಚರ್ಗೆ ಅಂತ ಅಂದರೆ ಅದು ಜರ್ಮಿನೇಷನ್ ಆಗಬೇಕಾದ್ರೆ ಈ ಸೀಡ್ಲಿಂಗ್ಸಲ್ಲಿ ಸಸಿಯಾಗಿರ್ತದೆ ಸೀಡ್ಲಿಂಗ್ಸ್. ಸೊ ಅಲ್ಲಿ ಮೆಚುರಿಟಿ ಆಗಿರೋದಿಲ್ಲ, ಅದು ಕ್ಲೋರೋಪ್ಲಾಸ್ಟಲ್ಲಿ ಪೂರ್ತಿ ಫಾರ್ಮೇಷನ್ ಆಗಿರೋದಿಲ್ಲ. ಅವಾಗ ಅಲ್ಲಿ ಏನು ಬೇಕಾಗುತ್ತಪ್ಪ ಅಂದರೆ ಫುಡ್ ಬೇಕಾಗುತ್ತೆ, ಹೌದಾ? ಸೊ ಅದಕ್ಕೋಸ್ಕರ ಅದು ಎಂಡೋಸ್ಪರ್ಮ ಪ್ರಿಸರ್ವ್ ಮಾಡಿ ಇಟ್ಕೊಂಡಿರುತ್ತೆ. ಆದರೆ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ಸಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇಲ್ದಲೆ ಅದು ಹೆಂಗೆ ಸೀಡ್ಲಿಂಗ್ಸ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗುತ್ತೆ ಅನ್ನೋದು ಒಂದು ಕ್ವಶನ್ ಇರ್ಬೋದು. ಅಲ್ಲಿ ಏನಪ್ಪ ಅಂತಂದರೆ ಕಾಟಿಲೆಡನ್ಸಿಂದ ಏನು ಮಾಡ್ತಂದ್ರೆ ಫುಡ್ನ ತೊಗೊಳುತ್ತೆ. ನಮಗೆ ಗೊತ್ತಿದೆ ಗ್ರೌಂಡ್ ನಟ್ ಪೀನೆಲ್ಲ ಕಾಟಿಲೆಡನ್ಸ್, ನಾವು ಕೂಡ ಕಾಟಿಲೆಡನ್ಸ್ನ ತಿಂತೀವಿ. ಸೊ ಅಲ್ಲಿ ಫುಡ್ಡು ಕಾಟಿಲೆಡನ್ಸ್ ಟೈಪಲ್ಲಿ ಇರುತ್ತೆ. ಓಕೆ, ಈಗ ನೋಡಿ ಎಕ್ಸಾಂಪಲ್, majority of monocots like cereals, okay, example paddy, ragi, wheat, maize. ಸೊ ಇದರಲ್ಲೆಲ್ಲ ಇರೋವಂಥದ್ದು ಏನಪ್ಪ ಅಂತ ಅಂದರೆ, ನಾವು ತಿನ್ನೋವಂಥದ್ದೆಲ್ಲ ನೋಡಿ ಪ್ಯಾಡಿ, ರಾಗಿ, ವೀಟರೆಲ್ಲ ತಿನ್ನೋವಂಥದ್ದು ನಾವು ಕಾರ್ಟಿಲೆಡನ್ಸ್ಗಿಂತ ಜಾಸ್ತಿ ಆದಪ್ಪ ಅಂತಂದರೆ ಎಂಡೋಸ್ಪರ್ಮ್. ಓಕೆ, ಸೊ ಹಾಗಾಗಿ ಇಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಪ್ರಿಸರ್ವ್ ಆಗುತ್ತೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಟ್ರಿಷನ್ಸ್ನ ಸೀಡ್ಲಿಂಗ್ಸಿಗೆ ಕೊಡುತ್ತೆ, ಸಸಿಗೆ ಕೊಡುತ್ತೆ. ಅದರಲ್ಲಿ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ಸಲ್ಲಿ ಕಾರ್ಟಿಲೆಡನ್ಸ್ನ ಕೊಡುತ್ತೆ. ಅದು ಕಾಂಪಿಟೇಟಿವ್ ಕೇಳ್ಬೋದು, ಸೀಡ್ಲಿಂಗ್ಸ್ಗೆ ನ್ಯೂಟ್ರಿಷನ್ಸ್ ಯಾವುದರಿಂದ ಬರುತ್ತೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಕಾರ್ಟಿಲೆಡ್ ಈ ಥರ ಆಪ್ಷನ್ಸ್ ಕೊಟ್ಟಿರ್ತಾರೆ. ದಟ್ ಈಸ್ ಫ್ರಮ್ ಕಾರ್ಟಿಲೆಡನ್ಸ್ ವೇರ್ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್. ಬಟ್ ಇನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್, developing embryo also utilize the endosperm and the preserved endosperm supplied for developing seedlings when it is in immature condition. And also coconut, ನಾವು ನಿನ್ನೆ ಡಿಸ್ಕಷನ್ ಮಾಡೋದಕ್ಕೆ ಬೇಕಾದ್ರೆ ಹೇಳಿದ್ವಿ ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರುತ್ತೆ ಅಂತ. ಕೋಕೋನಟ್, ದೆನ್ ಗ್ರಾಸ್ ಎಂಬ್ರಿಯೋನ ಡಿಸ್ಕಷನ್ ಮಾಡಿದ್ವಿ, ಗ್ರಾಸ್. ಸೊ ಇದರಲ್ಲೆಲ್ಲ ಏನಪ್ಪ ಅಂತಂದರೆ ಇದಿರುತ್ತೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಇರುತ್ತೆ. ದೆನ್ ಹಾಗಾದರೆ ಡೈಕಾಟ್ಸಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ ಯಾವುದು? ಡೈಕಾಟ್ ಅಲ್ಲಿ ರಿಸಿನಸ್ ಆರ್ ಕ್ಯಾಸ್ಟರ್ ಅಂತ ಕರಿತೀವಲ್ಲ, ಹರಳೆಣ್ಣೆ ಗಿಡ ಅಂತ ಕರಿತೀವಲ್ಲ, ಕ್ಯಾಸ್ಟರ್ ಆಯಿಲ್ನ ಎಕ್ಸ್ಟ್ರಾಕ್ಷನ್ ಮಾಡ್ತಾರಲ್ಲ. ಸೊ ಅದರಲ್ಲಿ ಏನಿರುತ್ತಪ್ಪ ಅಂತಂದರೆ ಆ ಡೈಕಾಟ್ಸಲ್ಲಿ ರಿಸರ್ವ್ ಫುಡ್ ಮೆಟೀರಿಯಲ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರುತ್ತೆ. ಸೊ ಕೆಲವೊಂದು ಡೈಕಾಟ್ಸಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇರುತ್ತೆ, ಲೈಕ್ ಕ್ಯಾಸ್ಟರ್. ಮೆಜಾರಿಟಿ ಆರ್ ಡೈಕಾಟ್ಸ್ ಆರ್ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ಸ್. Like that, majority of monocots are endospermic seed, and some monocots are also having they does not contain endosperm. And those are in category of non-endospermic seed. That is example orchids. Okay. So far we discussed about the fruit, seed. Before that we studied the embryogenesis. In next video we will discuss about significance of seed and other aspects. Take down. Thank you.